The axial skeleton includes the skull, the vertebral column or the spine, the sternum, and the ribs. Skull, vertebra, sternum, and the ribs. The source of the axial skeleton either mesoderm or ectoderm, which is the neural crest cells. The very source is the paraaxial mesoderm, which forms the neurocranium, and the spine, the vertebral column. The neurocranium is a part of the skull that embraces the brain. The lateral plate mesoderm, the second source, and this forms the limbs. Lateral plate mesoderm from the parietal layer or somatic layer of lateral plate mesoderm forms the limbs. The neural crest cells form the viscerocranium, which is the face, the facial bones come from the neural crest cells. Here we can look to development of an embryo through gestational day 16, 17, 21, and 24. In the gestational day 16, we have the amniotic cavity, the yolk sac cavity, and the epiblast, and the formation of mesoderm and endoderm. The epiblast differentiates into these three layers. Gestational day 17, we have ectoderm, mesoderm, and endoderm. And in the mesoderm, we have start of differentiation into three parts: paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. In the gestational day 21, we have the ectoderm and also the mesoderm. The paraaxial mesoderm forms the somite, the intermediate mesoderm, and the lateral plate mesoderm, which differentiates into two layers. You will see in the gestational day 24 here more differentiation of the mesoderm into somite, intermediate mesoderm, and the lateral plate mesoderm is differentiated into two layers: somatic lateral plate mesoderm and visceral lateral plate mesoderm. And we have said that the paraaxial mesoderm or the somites form the neurocranium and the vertebral column or the spine, and the somatic layer of the lateral plate mesoderm or the parietal layer form the limbs, and the neural crest cells, they form the viscerocranium or the face. The neural crest cells, we can explain it in this diagram into a fetus or an embryo of 22 days. We have a look from above. This is the region of the head or the cranial part, and this is the caudal part, the tail. And here we have the somites, these rounded structures, the somites. If you do a cut edge or transverse section here, you will see this image on the right side. The neural tube is formed, and below the neural tube is the notochord, and above the neural tube we have the dorsomedially we have the neural crest cells, and above here we have the surface ectoderm. Furtherly, with further development, we'll see the neural crest cells, neural tube, notochord, and the somites are more differentiated, the intermediate mesoderm and the lateral plate mesoderm. And the neural crest cells, they form the facial bones, the face, and part of the cranial vault, and also part of the membranous part of the skull of the chondrocranium. Let's have a look about on the development of the skull. And we have two parts of the skull, neurocranium and visceral cranium. The neurocranium is a part of the skull that embraces or contains the brain. The neurocranium has two parts: membranous part, which ossifies by intramembranous ossification, so it's called membranous part, is the cranial vault. Okay, and the cartilaginous part called chondrocranium, which is the base of the skull, and is formed by endochondral ossification or cartilaginous. By cartilaginous, we mean that the mesenchymal cells, they condense to form cartilage, and then the ossification centers later appear to form the bone. At first, they start as hyaline cartilage. The chondrocranium is the base of the skull, and also part of the chondrocranium is formed by the neural crest cells, and the posterior part, posterior to the pituitary fossa, is formed by the paraaxial mesoderm from the skeletal part of the somite. The second part of the cranium or the skull is the visceral cranium or the face. The facial bones, frontal, nasal, lacrimal, maxilla, mandible, part of the sphenoid, part of the temporal bone, squamous temporal bone, the hyoids, they come from the neural crest. Neural crest is the source, and they come from the first two pharyngeal arches. So this is about brief about the development of the skull. The paraaxial mesoderm, as it appears in this image, this is the neural falls. Later on, it closes to form the neural tube, and below the neural falls, we have below the neural tube is the notochord. The somites, as it appears here, on each side of the neural tube. Then it further differentiates into dermatome, skeletal, and dorsomedial muscle cells and ventrolateral muscle cells. So this is a somite in image A. With further development, the cells on the ventromedial wall lose their epithelial arrangement and they become mesenchymal to form the skeleton. Cells in the ventrolateral and dorsomedial regions, ventrolateral and dorsomedial regions, they form muscle cells, ventrolateral muscle cells, dorsomedial muscle cells, and also migrate beneath the remaining dorsal epithelium, which is the dermatome, to form the myotome. So the dorsomedial muscle cells and the ventrolateral muscle cells, they form the myotome, and this is dermatome, and this is the skeleton. These are the different parts of the somite or the paraaxial mesoderm. Let's now talk about the development of the vertebra. The vertebrae forms from the skeleton part of the somite, and the typical vertebra has a vertebral arch, vertebral foramen, vertebral body, transverse processes, and spinous process. During the fourth week, the skeleton cells migrate. These skeleton cells migrate around spinal cord and the notochord to merge with the cells on the opposite somite, opposing somite on the other side, the neural, on the other side of the neural tube. With further development, the skeletal portion of each somite undergoes what is called resegmentation. What does we mean by resegmentation? We have a look here in the longitudinal section. The skeleton undergoes resegmentation. We mean that the caudal half of each skeleton grows into and fuses with the cranial half of the subjacent skeleton. This is the skeleton and this is the other, this is third and this is the fourth. Here is the myotome and here the intersegmental arteries, here this spinal nerves, this is the myotome. The skeleton has two parts or half, cranial half جزء اللي فوق والcaudal half جزء تحت. The caudal half of each skeleton merge with cranial half of the lower or the subjacent skeleton. Together they fuse to form the vertebra or the vertebral body. So each vertebra is formed from the combination of the caudal half of skeleton and the cranial half of the subjacent or lower skeleton. Then we have the intersegmental mesenchyme. This intersegmental mesenchyme does not differentiate and remains to form the intervertebral disc. So the image in A, this occurs at the 4th week. In B, in further development, there is proliferation of the caudal half of one skeleton into the cranial, here into the cranial part or cranial half of the subjacent skeleton. So, and note the appearance of the intervertebral disc. This nucleus bulbosus is formed by the notochord, surrounded by the annulus fibrosus. This is the annulus fibrosus circular fibers to form the intervertebral disc. In C, these are the vertebrae, which are formed by upper and lower halves of two successive skeletons. We have the intersegmental tissue form the intervertebral disc. We have here the myotomes, and these are the segmental spinal nerves. Although the notochord regresses entirely in the region of the vertebral bodies, it persists and enlarges in the region of the intervertebral disc to form the nucleus bulbosus, and it's surrounded by, as we said, the annulus fibrosus circular fibers. Combine these two structures, the nucleus bulbosus and the annulus fibrosus, form the intervertebral disc. Here we'll talk about the development of the sternum in early sternum and ribs. We start with the ribs in early week 8. The ribs posterior, this is a cross section or transverse section through the thorax. You have the heart, lungs. Posteriorly, this is the rib primordium with tubercle of the rib. In week 12, the rib primordium will have a costal ossification center, and we have rib head and rib tubercle attached to the vertebra. For the sternum, we have early precartilaginous stage, late precartilaginous stage, and cartilage stage, and then at birth. The precartilaginous stage or mesenchymal stage, in week 6, we have two sternal bars and suprasternal masses, here 3, and the ribs also are formed in the mesenchymal stage. In the late precartilaginous stage, the two sternal bars fuse up here with the suprasternal masses, and the upper ribs fuse with the sternal bars. In the cartilage stage, the sternum is now cartilaginous, and is attached to most of the ribs, and the clavicle is ossified in week 9. The clavicle starts also in the mesenchymal stage, and it ossifies and the ossification center appears at week 7, becomes ossified at week 9. At birth, the sternum has its shape, still cartilaginous with ossification centers appears. The xiphoid ossification center appears at 3 years. The ossification centers for the sternum, manubrium, and the upper part of the body appears at 5 to 6 months. And we have the ribs ossified laterally, and the costal cartilages are here attached to the sternum, to the cartilaginous sternum, which is the sternum that contains ossification centers. In the adult, we have ossified adult manubrium, body of the sternum, and we have the xiphoid low down, which is cartilage in early youth. Later on, it becomes ossified, and but it is cartilage in early youth. This is the adult shape of the sternum, and we have here the costal cartilages, and also the joints disappear puberty to 25 years. At puberty, up to 25 years, the joints that were present between the parts of the sternum disappears. Now we will move to some clinical correlates related to the development of the axial skeleton. Firstly, we have the craniosynostosis, which is a group of abnormalities that are caused by premature closure of one or more sutures. And as we know, the sutures, the anterior fontanelle closes about 3 months after birth, and this is the posterior fontanelle closes about 3 months after birth, and the anterior fontanelle closes around the middle of the second year. And then many of the sutures disappear during the adult life. If one of the sutures or more closes earlier than the expected time to close, to result in abnormalities in the shape of the skull, and this is called craniosynostosis. For example, we have the scapucephaly. Premature closure of the sagittal suture will result in scapucephaly. Long and narrow skull with frontal and occipital bossing. This is results from a premature closure of sagittal sutures called scapucephaly. This is one of the craniosynostosis abnormalities. Second one is brachycephaly, and this is due to premature closure of the coronal and lambdoid suture. Third one is plagiocephaly, due to premature closure of coronal and lambdoid suture of one side. Let's have a look on the image. here. This is the brachycephaly, short skull, due to premature closure of the coronal suture and lambdoid suture posteriorly, resulting in short skull. So, skull brachycephaly. In B, we have the plagiocephaly. Plagiocephaly, it results from premature closure of coronal and lambdoid suture on one side. Coronal and lambdoid suture on one side, okay, closed earlier than expected, resulting in what is called plagiocephaly. This is CT scan, 3D CT scan for this baby with plagiocephaly. We notice the premature closure of coronal suture and lambdoid suture on one side, resulting in plagiocephaly here with bossing of frontal region on one side, on the opposite side, okay, and this is how it looks. This is called plagiocephaly, premature closure of coronal and lambdoid suture on one side, resulting in this shape, bossing of the skull from one side. Here we have the vertebral defects, and we have the cleft vertebra or the spina bifida. When the vertebral arches fails to close and embrace the spinal cord, they remain like this, open, just covered by skin, and this is called spina bifida occulta. We find hair on the skin. This is a characteristic for spina bifida occulta, where there is no involvement of the neural tube or the spinal cord. The spinal cord is intact, but the vertebral arches fails to close. On the right side here, we have a female child with spina bifida occulta, where there is here a tuft of hair in the lower lumbosacral region, usually occurs at the lower lumbosacral region, and this is called the spina bifida occulta. It is one of the varieties of the spina bifida. Later on, we have other varieties. We will discuss it in the nervous system, the development of the nervous system and defects of the neural tubes. We have other types. We will discuss it later in the coming lectures. So this is the end of our lecture. We have discussed the development of the axial skeleton regarding the skull, the vertebral column, and the sternum and the ribs. And we have mentioned some of the clinical correlates related to the skull development and the vertebral development. Thank you for your listening.